Okay, um, thank you and welcome everyone for today's SLAM seminar. It's a great pleasure to invite and introduce Babak Vajdi Hokmabad, um, who who's our speaker today. He's an associate research scholar in chemical and biological engineering at Princeton University, currently working with Professor Charles Schroeder and previously worked also as a postdoc with Sujit Datta, who's currently now at Caltech. Babak earned his PhD at the Max Planck Institute for Dynamics and Self-Organization. His research focuses on the physicochemical hydrodynamics of active and living systems, and his work sort of integrates soft matter physics, fluid dynamics, and microbial biophysics in with very, very fascinating consequences, some of which I believe you'll hear about today. He studies how synthetic and biological active matter, ranging from self-propelled emulsions to microbial communities and complex media, generate and respond to mechanical cues, and is very keen on trying to use these platforms to reveal new principles of self-organization in structured environments. So thank you, Babak, and take it away. I will spotlight you now for everyone, and everyone in the audience, feel free. Babak has kindly agreed to be open to questions throughout the talk. We will also
Yeah.
have questions at the end. And so thank you again. Take it away.
Thank you very much. Yeah. Hello, everyone. So today I want to talk about two projects that I've been busy with during my postdoc at Princeton University. So I want to talk about metabolism, motility, and mechanics, and how microbes use these factors to be able to thrive in complex environments, to develop strategies to live and proliferate in complex environments. So when we think about active and living systems, from a physicist point of view, these are usually a collective of active agents that are driven out of equilibrium because they are locally at the microscale consuming energy. The typical examples that I am personally interested in are microbial organisms and synthetic active particles. These are essentially like chemical micromachines that consume nutrients and chemical fuel in the environment, generate energy, and also produce some chemicals that later on they secrete into their environment, a process known as metabolism in microbial organisms. So these biological and chemical activity, this kind of biological and chemical activity can be manifested in the form of microbial growth, as you can see here in this top video on the left. But it could also manifest itself in the form of generation of mechanical work. For example, we know that many microbes are able to self-propel in the environment using these appendages, these red appendages that are known as flagella, or synthetic active particles are able to self-propel in certain environment. In this case, a chemically active droplet is driven by Marangoni stresses on its interface in a surfactant solution. So these systems have been studied extensively, right? But typically these are studied in uniform, homogeneous, and idealized environments, which are starkly different than what you expect would happen to them in their natural habitats. In their natural settings, these systems are typically interacting with complex environments. Examples are like the pore space of the soil, where you have complex geometries, chemical heterogeneities in the form of chemical gradients. We know that microbes are able to sense gradients in their environment and then respond to that, a process known as chemotaxis, and also mechanical heterogeneities in the environment, as you can see here in this stratified porous medium. So the goal of my research is to understand this interaction between biological systems and their complex environment and see how new behavior emerges, right? But in our quest to understand such dynamics, there's a missing piece. Many of these systems, like think about the soil or the airways in our lungs, we do not have direct visual access to be able to observe the dynamics in a space and in time, right? As a result, we don't really know how this feedback between activity and the environment works. That's why in my research, I try to build biomimetic environments that are also transparent. And then we embed the living system, the active system, in such complex biomimetic environments, and then look at the dynamics in space and time. And we are able to decipher the mechanochemical coupling between the biological system and the physical and chemical properties of such environments, which leads to understanding the emergent dynamics that emerges because of such complex couplings. So today I will talk about two specific systems. The first one is about microbial proliferation and dispersal in complex yield stress environments, the one on the left. And second, I will talk about microbial self-organization driven by motility and metabolism and bacterial suspensions. So in the first project, metabolism and growth are coupled to complex geometries and chemical gradients and heterogeneous mechanical heterogeneity, basically. And we call it biogenic bubbles enable long-range microbial dispersal. When we think about microbes, we know that they have developed strategies to be able to be motile, to swim around in the environment and perform tasks such as chemotaxis, foraging for food, and also use that as a navigation, for example, as a navigation strategy to boost range expansion, right? But not all microbes are motile. A familiar example for all of us is yeast. Yeast cells can be found in food items and biological tissue where they cause infection. And most importantly, you can find similarly immotile microbial organisms within the ocean sediment. These organisms are known as methanogens, and then they produce methane in the ocean sediment, and with that, they during anaerobic processes, and then they contribute to the cycle of methane in the ocean, and then later on into the atmosphere. So when you think about these environments that I mentioned, the sediment and soil, food items, biological tissues, these are typically complex yield stress, crowded environments. And we asked what would happen to a colony of immotile cells where they are when they are confined in such environments. Do they locally get trapped and then stay there for a long time, or do they find a way to break out of such microenvironments to be able to disperse over long ranges? And today I'll show you that they find a way, and that's not using motility, but by using the basic principles of fluid mechanics. Okay, so let's move forward. Okay. The model the model system that we use are the famous Baker's yeast, which are single-cell immotile fungus. And since those environments that I mentioned are basically not transparent, we use transparent granular hydrogel, a suspension of these microgels, basically, as a model system for sediments, for complex yield stress environments. We make a dense suspension of these hydrogel particles. Each particle is about 5 to 10 micron in diameter. And if you look at the mesh size within each hydrogel particle is about 100 nanometers. So small molecule nutrients and other metabolites can freely diffuse within the matrix. Since yeast cells are like 5 to 10 microns in diameter, if you put an inoculum of cells, a colony of cells within the matrix acts as some sort of continuous gel with respect to the colony. And that means that by changing the concentration of the particles, okay. By changing the concentration of particles, we can tune the mechanical properties of this matrix. Here we can see by increasing the carbopole concentration, first of all, we observe yield stress behavior, which means at low shear rates, the material is acting like a solid, but at higher shear rates, it is acting as a liquid. And by changing the concentration of carbopole gel that we are using, we can achieve a broad range of yield stress values. So the material can become stiffer by increasing the granular concentration. Okay, so let's just start from the experiments. Here at the bottom right, you see an initial inoculum of yeast cells that we embed inside the granular hydrogel, and the darkest space is basically the matrix, the hydrogel matrix. It is dark because it is transparent. So what we expected for this colony was that, as our lab has previously shown, the colony should stay there, and it should slowly grow because we've shown that nutrients and other metabolites can only penetrate up to like 10 to 20 microns into the colony. So the growth is limited to the surface of the colony, and the bulk is not growing because it is depleted from nutrients and oxygen. So what we expected was slow surface limited growth and an isotropic growth in all directions. But if you give enough time to this colony, what you observe is this. So the colony has grown anisotropically in the vertical direction all the way to the top air gel interface. And if you run the scale bar at the bottom, you see that that is 5 millimeters, right? So this colony that I'm showing you is about a few centimeters, which is surprising. So we thought the only way that the colony could change the matrix, its environment, since these are immotile, is through its metabolism. And we know that we have glucose in the environment, and that means these yeast cells are performing fermentation. So to test whether this effect, this directional growth is because of fermentation, we switched to a non-fermentable metabolite, and that is glycerol. And then we observed what we initially expected. The colony grows very slowly. It's a surface-limited growth, and it is isotropic in all directions. Okay, so fermentation has something to do with this. So a quick update on like a reminder about fermentation. That basically means the yeast cells are consuming the glucose in the environment and break it up into ethanol and carbon dioxide. Now, if I zoom in onto the yeast inoculum, you see that since carbon dioxide is being produced, gradually the concentration of carbon dioxide that is dissolved in water increases, reaches to the saturation point, and at that point, a bubble of carbon dioxide is nucleated. It grows. Once it is large enough, it starts to yield the hydrogel matrix. It starts to rise, and as it rises, very slowly, it carries the colony in its wake. Now, do you have evidence for what I just suggested here? To provide evidence,
So, Babak, I have a quick question.
Yeah.
Just with regards to scales, the granular, so the inoculum is much, much bigger than the size of the granular beads. Is that correct?
Yes,
And
it's
so
millimeter scale, yeah.
and the size of the carbon dioxide bubble as well at nucleation is bigger.
Yeah, it is. At nucleation, it is very small,
Okay.
but it cannot move because it is stuck within the matrix.
Okay.
It can only move when the buoyancy of the bubble is large enough to impose a force strong enough to yield the matrix, to fluidize the matrix basically locally, and then it can move.
How does it overcome does it wet the surface? Do you do anything to prevent the bubble from wetting?
Yeah, so since we're using hydrogel, which is like 99% water, you can assume that this system is like water in terms of surface tension. But if you switch to something that is wetting, like I don't know, soil, actual soil, then you might expect different behavior in terms of bubble dynamics. Yeah, that's a great point.
Okay, so you don't have any issues where the bubble sort of gets trapped in some tortuous pores, but it actually does yield and push out the beads. Okay.
Yeah, yeah. Okay, so to show whether this is actually a carbon dioxide, we used the pH-sensitive fluorescent dye, and then we performed confocal imaging at this cross-section, the red cross-section. And then we know that dissolved carbon dioxide reduces the pH locally, right? And we can capture that gradient in pH and carbon dioxide concentration. And then we can measure the carbon dioxide concentration from the pH. And if you look at this chymograph, you see that the concentration of carbon dioxide gradually increases at the location of the colony, and then it reaches to the saturation point. And that's more or less when the bubble is nucleated and it starts to grow. Okay. We also did a very simple simulation of basically a reaction diffusion where the colony consumes glucose in the environment and then secretes, produces carbon dioxide, and then the carbon dioxide diffuses. And we see that very quickly the concentration at the location of the colony reaches to the saturation point, which further supports that supersaturation happens and the bubble is nucleated. And the nucleated bubble is a carbon dioxide bubble. Okay, so now we know the system is driven by a carbon dioxide bubble. We asked what sets the shape of the colony, and is it a function of the morphology of the bubble? So when we look at the growth dynamics of the bubble, we see there are three regimes. First one is nucleation and spherical growth. Then it switches to, without moving, it switches to asymmetric growth. So the bubble becomes elongated. And then once the buoyancy of the bubble is large enough, it is able to yield the matrix in its perimeter around it, and it starts to rise. So we can characterize first the rising dynamics with a non-dimensional number that is known as Bingham number, which is the ratio of yield stress to buoyancy stress. There we see that the Bingham number gradually decreases as the bubble is growing in time. And then once it reaches to a critical value, which is 0.3, and this is comparable to the literature value, then the bubble starts to rise. So this critical value is giving us one characteristic that we can test in our future experiments. And we can characterize the shape of the bubble using the bond number, which is the ratio of buoyancy forces to the capillary forces. And there we observe the bond number increases rather quickly, and then once it reaches to a value, to a plateau that is close to bond number equal to 1, then the bond number does not really change. It stays the same. And we have used the radius of the bubble, not the height of the bubble, to calculate the bond number. So this gives us two hypotheses to test, right? The critical Bingham number is able to predict the onset of rising, and bond number equal to 1, which technically corresponds to the capillary length of the system, the natural capillary length of the system, is able to predict the radius of the bubble. And the bubble and the growth continues until the bubble reaches to the bond number equal to 1, more or less. To test these, we change the yield stress of the environment, increase the yield stress by 1 order of magnitude, and then we measure the critical Bingham number at the onset of rising, and we observe that we always get the same value, 0.3, which confirms our hypothesis. And when we look at the bond number, we see it increases gradually, but it is going to basically it plateaus at a bond number that is close to 1, which again corresponds to the capillary length of the system. And this is reflected in our measurements of the actual dimensions of the bubble that we have in the system. The height of the bubble increases because we need buoyancy force to be able to yield the matrix. But the radius of the bubble only increases until it reaches to the capillary length of the system. And after that, surface tension is not strong enough to keep the bubble spherical. As a result, it grows in the vertical direction, which is basically a direction of the buoyancy force. Now, why is this important? When you look at the radius of the columnar colony, this colony of cells, you see that basically the colony radius is very similar to the radius of the bubble. And what does this tell you? It tells you that the capillary length of the system is controlling the radius of the bubble, and the radius of the bubble is controlling the radius of the colony that has been formed because of the motion of the bubble. Okay, so now we understand the dynamics of the bubble, but what drives this entrainment, basically? What moves the colony upwards? And we know that the cells are not adhering.
Sorry, another quick clarification regarding the definitions on your previous slide, regarding the definitions of the Bingham number and the buoyancy number. Sorry, the bond number. So the Bingham number, so you use the height of the bubble to estimate the buoyancy stress. Can you say a bit more why is the height? Because I would have thought that it's the highest curvature, which will be controlled by the radius of the bubble that would apply the pressure needed to yield the material.
Yeah, so you should use the highest curvature basically when you have surface tension involved, right? In this case, what is competing is yield stress, of course, and the buoyancy stress, the buoyancy force that is trying to yield the matrix. And typically in the literature, what people do is they calculate some sort of equivalent radius when the bubble is not spherical. We tried that, uh, and that gives you, I want to say, non-universal numbers in terms of critical Bingham number. And we thought basically what the shape that we have is like a cylinder, and then the buoyancy force scales with the height of the bubble,
Okay.
right? And that, or basically the volume, we calculated the volume of the cylinder, and you're left with the height within the estimate for the Bingham number, and then you get this straight line, this flat line for the critical Bingham values.
Okay.
But I think there's another question as well.
Yes,
Yeah.
Dimitrios, go ahead.
Hi, thanks. That's very interesting. I have a quick question about your previous slide and maybe a general question. So, is there a single bubble forming? So, the previous slide 16.
Yeah.
Is there a single bubble forming always, or is there a nucleation that happens, in various sites and then the bubbles merge? Because here it seems like this. And if that is true, the latter, when can I start treating this as a single bubble?
Yeah, that's a great question, and I will actually answer that within the next few slides.
Okay, thanks.
Yeah, but that's great insight, actually. Yeah. So I will move forward, and then we'll get to your question. Yeah. So what causes the colony to be carried by the bubble? When you look at the classic literature, there's this concept, Darwin's drift, which means basically when one object is moving inside a fluid, its motion also carries material in the form of liquid with itself, right? As a result, you get fluid, a volume of fluid that has been moved because of the motion of this object. Now, there's a analytical solution for that in the Stokes flow. However, our system is a complex yield stress viscoplastic fluid, right? So we couldn't really come up with an analytical solution. That's why we asked our collaborators at the University of Amsterdam to do simulations, like DNS simulations for a bubble rising in a viscoplastic fluid. We used the basic Bingham fluid model, which is incompressible, and there's no elasticity involved, which could be important, but not necessarily for this specific application. So here you see the result of the simulation. You see the bubble on the right-hand side, that you see the deformation field. The bubble locally yields the matrix around it, and fluidizes the matrix around it, and it is able to deform this imaginary blue line and carry material along its motion. And it's the amount of fluid that it can carry. It's a function of the Bingham number. So if you go for lower Bingham number, you can carry more fluid forward. And this basically tells us that hydrodynamic entrainment is enough for the colony to be carried behind the bubble and form this kind of columnar structure. And there has been reports of similar hydrodynamic drift in nature, not in viscoplastic, of course. Biogenic ocean mixing by Darwin's drift has been shown to be effective in inducing considerable mixing in the ocean. And there's this nice paper from Arnold Matthijsen's lab where they show collective entrainment by a school of microswimmer is enhanced because of collective motion in confined spaces. So it's a very useful tool to look at the material transport in active motion and in biogenic systems. So going back to the simulations, there's one caveat here. when you look at the deformation of the matrix around the bubble, and if you add tracers which represent the yeast colony, you see that this volume gradually tapers, and then eventually the last yeast cell loses its contact with the bubble. But what we observe in our experiments is that the colony is always a cylindrical columnar structure, which goes all the way to the top surface. So why is that? And now I want to answer Dimitri's question. Here I have increased the yield stress from left to right by 1 order of magnitude. And there's a lot happening here. I want to say this is a dream for a fluid mechanics person. But when you look at all of these systems, you see that there's always more than one bubble, because the colony is constantly producing carbon dioxide, and that leads to nucleation of multiple bubbles. And they, more or less all of them, follow the same track, because the first one is inducing some sort of history in the environment, which could be either mechanical or rheological, basically. And this sequential rising by the bubbles enables enough volume transport, volume drift, that ensures the motion of the colony all the way to the top surface. And this has been corroborated in our simulations, where we add 4 bubbles, and then we observe the entrain volume in time, and we see this stepwise addition, this stepwise increase in the entrained volume, which ensures the formation of the columnar structure. Okay, so
maybe it's a useful point to ask a question from the chat.
Yeah.
Yasha Kresh asks, regarding the bubble morphology, is the bubble shape controlling the colony morphology, as you mentioned, wherein the size, the radius of the colony that's entrained is comparable to the radius of the bubble? Or do you think that there's some aspects of the reverse, wherein the colony, by virtue of adhesion, affects the shape of the bubble itself?
No, so this specific strain of yeast cells do not adhere to the interface. There are strains that you can choose that would adhere to the surface, and then potentially, if you let them grow on the interface, there's a regime where they cover up the surface, and then they will change the interfacial behavior of the bubble. In this case, they do not adhere to the surface, and it's the motion of the bubble that determines the shape of the colony. Yeah, it's basically because of the hydrodynamic entrainment. But great question. Is there another question that?
No, not that I can see now.
Yeah. Great. So we understood the mechanism for the formation of the colony. Then we asked, what would happen if you put 2 colonies that are neighboring, like next to each other? How would they interact? And before showing you this video, I want to give you some hints. What I did was I added some yeast cells to a container full of hydrogel matrix. And then we let it sit for 0 days, 1 day, 2 day, 3 day, and 7 days. And then after the fermentation basically was done, we measure the pH of the matrix, and we measure the yield stress of the environment. And we observed that because of the presence of the products of fermentation, carbon dioxide and ethanol, the matrix becomes softer, so the yield stress decreases, right? So if I now go back to this video, we can again do confocal imaging at this cross section, and then we see that carbon dioxide is being produced, pH is being decreased, and then there's a point in time where the clouds of carbon dioxide are overlapping between the 2 colonies, right? So with some calibration, what we did was we visualized and measured the local yield stress of the matrix based on the data that we collected with the confocal microscope. And you see, by increasing time, the space between the 2 colonies experiences a lower yield stress. So it becomes softer compared to the outside region. And that leads to some sort of rheological heterogeneity. There's a gradient in the yield stress of the environment. And it's been shown that in the literature, the bubble always chooses the path with a lower yield stress, so the softer space. And now you see when I play the video, the bubbles are deviated towards each other. This is, of course, partly because of the hydrodynamic interactions between the bubbles as well. But we've had cases where there's only one bubble and one columnar structure, and then the bubble is also deviated towards that. That tells you that this heterogeneity, this gradient in yield stress, leads to the deviation of the bubble. Now, this has consequences. It has implications. First one is, I want to say, biological, because if you look at the growth of colonies on an agar plate in 2D, you see that there's an effect called genetic demixing in 2D colonies, which has been well studied. You get basically from one colony, you get two multiple colonies. But in this case, we get mixing of two distinct colonies because of the interactions of the bubble and the mechanical motion of the bubble. And we have mechanical implications. So here we start from 9 colonies at the bottom. And you see that gradually, as the bubbles are rising, they start to interact with one another. They are deviated towards each other, and they merge. And you see this conduit formation process by the motion of biogenic gas, by the migration of biogenic gas. So at the end, we start from 9, we are left with like 5 colonies, which are further away from each other. So the length scale is changing and is dictated by the interaction between the bubbles and the heterogeneity, the rheological heterogeneity of the environment. So now, I want to go back to the first image that I showed you about the ocean sediment. There has been reports of these streams of methane bubbles, which are like centimeters apart, tens of centimeters apart. And these are more or less permanent, which tells me underneath there are conduits that are basically venting, releasing methane, biogenic methane from the ocean sediment. And another example that I recently found, and it's really cool, thing is, you see these finger-like structures, which are like tens of centimeters, I believe, and microbial mats in ice-covered Antarctic lakes. And you see there are tiny oxygen or methane bubbles that have been generated and nucleated within the mat, and then collectively those bubbles raise the matrix and then lead to formation of these finger-like structures. And this is another example published in 2025, I believe. Great. So this is a cool system. I would say what we can do with it is, I want to say we are now observing a new kind of motion, a new class of active matter where individual cells are not moving, but their metabolic activity leads to motion. Now we have a playground. We can play with the metabolic rate when you compare it to diffusion. We can play with the geometric confinement. If you bring the wall closer, you would see that the bubbles are nucleated on the boundaries, not within the matrix. And you can also play with the physical confinement, like the yield stress. We saw similar example where we switch from fluidization to fracture. And if you go for faster production, you might get a transition from a cloud of bubbles, which can be dangerous because it's really all of them together release a lot of carbon dioxide or methane into the environment, or a fracture network. And then we can play with the metabolically driven flows and then look at its consequences in terms of mass transport and also microbial ecology, where we have antagonistic or mutualistic interactions between different strains of cells, between different species of cells. Okay, so with this, I want to go to the second part of the talk, where we have metabolism and motility, and the colonies basically confined in a geometry, in a confined geometry, and chemical gradients are also involved. And it's called a breath of fresh air, where we study the spatial self-organization of confined bacterial suspensions. We know that microbes often inhabit confined, nutrient-limited spaces. This could be microbial communities in the soil and sediment, or the microbial communities within the host tissues and gels, like in the airway, in the colon. So we wanted to see how colony of motile cells respond to this. So today I want to talk about the interplay between confinement, nutrient transport, motility, and microbial metabolism, and how that determines the population self-organization. This could be very important because it influences the overall population functioning and the resilience to stressors, right? So the way they self-organize affects their response to, for example, antibiotics or phages and things like that, predators, for example. Okay, so the experimental setup is rather simple. We make a dense suspension of E. coli motile cells, and then we confine this between two glass plates. We sandwich it between two glass plates. The height of the confinement is 130 micrometers. The droplet diameter is millimeter scale, to a few millimeters, basically up to a few millimeters. Then we perform confocal imaging of the dynamics. Please mind that this means that the system is closed from top or bottom. It is only open at in the radial direction at these interfaces. Okay, so let's start from the experiments. If I start from a small dilute droplet, which means a low concentration of bacteria, this is what I observe. Basically nothing. Nothing really happens in time. And all cells are motile. Now, if I go for a large droplet, like a few millimeters in diameter, which is also very concentrated, the concentration of bacteria is really large, this is what I observe. The suspension spatially self-organizes into two phases. One dense one, and around it, the core is basically surrounded by an annular space. And then we see that the width of this annular space is more or less constant in time. And now if I go for a small droplet, which is concentrated, so the concentration of bacteria is really large, this is what we observe. The phase separation happens into the core and the annulus, but interestingly, this time, the core starts to shrink, and then eventually you'll see that it disappears. So the questions that we had was, why do the phases form? And then why do the phases sometimes disappear? To answer this, first we zoom in onto this region, onto the annular area. And interestingly, we observe that within the core, cells lose their motility. But within the annulus, and this is the droplet interface, within the annulus, cells are motile and they show flow patterns at high concentrations. They show active turbulence, basically. This could provide a hint for us that there's some sort of metabolic switch happening. And this is the cross-sectional image that we are observing, basically. And then when you look at the literature, you see that E. coli loses its motility after a few minutes of oxygen deprivation. So this could be because of oxygen. So to test this, we added a fluorescent probe for oxygen to measure the oxygen concentration. It is a little bit tricky because you have to encapsulate it in micelles because the dye molecule itself is a bit toxic for the bacteria. So you need to be careful about that. And then if I play the video, you see that here darker means more oxygen. Oxygen is depleted within the core. And there's a gradient from the interface all the way to the core. And then at the core interface, the oxygen concentration reaches to 0, and it stays like that. Okay. So how can we quantify this? First, we ask, how far does oxygen penetrate into the droplet? And by a simple balance of diffusion and consumption, by assuming quasi-steady state, you can get an estimate for the initial widths of the annulus at the beginning of the experiment, which is based on the diffused zero oxygen concentration at the interface. Kappa is the consumption rate of oxygen by the bacteria and cell concentration. And when we measure the initial widths of the annular by increasing the cell concentration, we observe that we are able to recreate this scaling from this simple calculation. Now, interestingly, so this is a hint that this is because of oxygen. Now, interestingly, if I switch from this medium, which is a minimal medium, media, to a rich media where there's an abundance of glucose and nutrients, the consumption rate decreases significantly by almost, I don't know, I think 1 order of magnitude or even more. And but we again see the scaling. This tells you that this is most probably because of the oxygen consumption and depletion, in particular in the rich media, because they need to metabolize the sugars, they consume more oxygen. Okay, so we understood why the phases form. Then we asked, why do the phases sometimes disappear? And to understand that, we will zoom in onto the interface of the droplet. And when we do that, we see that bacteria that are affected by the oxygen gradient, and they prefer to go towards the higher oxygen concentration, they start to accumulate, basically travel to the interface and accumulate at the droplet interface. Now, what kind of effect does that have? It basically enhances the gradient in oxygen concentration across the interface. And that means that the flux of oxygen that is coming inside the droplet increases because of this enhanced gradient. I cannot measure the oxygen flux. What I can do is I can measure the oxygen content, the overall oxygen content within the droplet. And we observe that after the core forms, the oxygen content gradually increases because of the enhanced flux of oxygen that is coming in. What happens is that this enhanced flux diffuses into the core. The cells that were sleeping, basically, in the core sense that there's new oxygen now. After a few minutes, they wake up, start to be motile. They join the active motile annulus, and then gradually this core shrinks in a matter of tens of minutes, and then it disappears. And once the core disappears, we reach the steady state, which looks like an aerobic state. And there's no core, but there's a little bit of gradient in oxygen concentration. So this is the full picture that we have now. That's the core, this is the active annulus, oxygen flux increases, and then eventually all the cells wake up and join the matrix. Okay, so by putting all these pieces together, we can get a minimal model for phase separation and dynamics. This is a continuum model where we model the bacteria dynamics. We have the undirected motion, which is the diffusion of cells, and we have the directed motion, which is the aerotaxis. Here, the aerotactic velocity is modeled using this aerotactic sensing function. It's like a drift, basically, that is biased towards a gradient in oxygen concentration. We also model the oxygen consumption and diffusion. So we have diffusion and we have cell uptake, oxygen cell uptake, which follows the Michaelis-Menten dynamics. We have the metabolic switch. So wherever there's no oxygen, oxygen is zero, motility, which is diffusivity of the cells, and chi, which is the aerotactic function, aerotactic coefficient of the cells, becomes zero, and cells stop to move. And we have this boundary condition where oxygen flux at the boundary is a function of the gradient of oxygen concentration at the interface. So with this, we get two important governing parameters, non-dimensional numbers, basically. The first one is the ratio of droplet radius to oxygen penetration lengths. This will tell us when a core will form. If the droplet is large enough so that in its core, there's a region where oxygen cannot penetrate in. And there's another non-dimensional number, KT tilde, which is the ratio of interfacial flux of oxygen to the oxygen transport rate. And if you equate the overall interfacial flux of oxygen to the overall uptake of oxygen, you'll get a scaling for this non-dimensional number as well. So now we have a criterion for core formation, the first one, and a scaling for the criterion for the core dissolution. So these are the experiments to just remind you what happens. A small dilute droplet, aerobic state, nothing changes. Large concentrated droplet, you get the core, and then the core shrinks a little bit, but it reaches to the steady state before the core disappears because the enhanced flux is not enough to provide the entire colony with oxygen. That's why the long-term steady state is anaerobic. And you have this state where you start from anaerobic and then you get to this aerobic steady state. Using the equations that I showed you, we did simulations and we observed that we are able to capture the dynamics in more or less good agreement. So the first regime is aerobic. The second one is permanent anoxia, which means the anoxic core forms and it does not disappear. In the steady state, you have anoxia state. And there's a transient anoxia state where you start from anoxia within the core, but then the core disappears, and then you're left in the steady state with the aerobic state, yeah. So now we go back to the governing parameters. We can generate a state diagram with R tilde and KT tilde to the power of -1. We add the criteria that I mentioned. So this is R tilde equal to 1. Below this, no core will form. Above that, cores will form, anoxic cores will form. And now this is the criterion for demarcating the permanent anoxia from transient anoxia. Above this line, the core forms and it stays. So you're left with permanent anoxia. So if we add simulation results, we see a good agreement, at least at larger values of KT1, KT to the power of -half, sorry. At lower values, there's a good explanation for this because this is more or less not really physical. The concentration is reaching to the maximum value, and that means very small droplet that is very concentrated. It is almost impossible. And if I now add the experimental, more than 250 experiments that we did, we get a very good agreement with the theoretical predictions and the simulations. And you see that we observe many examples of permanent anoxia as we predicted. So this basically provides quantitative principles to predict and control the phase dynamics when a colony of motile microbes are confined in a closed space. And the dynamics, as we showed, is mediated by confinement, metabolite transport, motility, and metabolism. So I want to end this part with this video. This is something that we're still thinking about. We wanted to look at the effect of viscoelasticity. And we know that viscoelasticity increases the swimming speed, and also it enhances the collective swimming behavior. On the left, you see the Newtonian case that I showed you before. The core forms, shrinks, and disappears. You get active turbulence within the active annulus region, but since the droplet is really large, it's hard to see. But if I now play the viscoelastic case, you see that very large scale vortical structures are forming, and then the gradient in cell concentration is enhanced. Still, you get the core formation and then core shrinkage and core disappearance, which is reassuring. But you see a lot of cool hydrodynamic effects, and these are things that we want to think about and explore further in the future. Okay, so with this, I get to the end of my talk. I tried to show you how active and living systems can couple to their complex environment and lead to new interesting phenomena. We talked about microbial proliferation and dispersal in complex yield stress environments, which are similar to the ocean sediment. And also I tried to show you when you add motility to the system, so responsiveness to chemical gradients, you get self-organization that leads to new structures, interesting structures, and adaptive behavior for suspensions of active bacteria. And I want to acknowledge all the collaborators on these two projects, Sujit, Mazi Jalal, Mira, Alex, Tom, Sebastian, and my undergraduate student, Ho, who's now a PhD student at MIT. Thank you very much for your attention, and I'd love to hear your questions. Thank you.
Thank you so much, Babak, for a fantastic talk.
Thank you.
We have some questions in the chat. We could start with that. And if people want to ask further questions, please feel free to raise hands. And okay, so there's a couple of questions in the chat. Let's start with that. Richard Gordon asks, for the first part of your talk, what happens in microgravity?
Oh, that's a great question. That's one of those, I want to say, kind of unnecessary cool experiments that could be done in zero gravity. So the bubble will not move, right?
Yeah.
Because there's no gravity. So what you get is this bubble that is growing and growing, and it'll push away the colony around it. Yeah. So I can't really think of a lot of cool things that could happen.
It sounds good. Well, maybe that'll be a next proposal to NASA.
I look forward to it, yeah.
Um, Shankar Lalita asks, what might explain the emergence of heterogeneity in the yield stress in a given hydrogel environment, seemingly appearing consistently between two nucleation sites? I believe this is also for the first part.
Yeah. So basically, each colony is producing carbon dioxide, and then the carbon dioxide is dissolved within the medium, within the aqueous medium. And since the dissolution of carbon dioxide reduces the pH in the environment, that leads to the decrease in the yield stress of the matrix. Basically, each individual hydrogel particle deswells a little bit, and then that means locally you get lower yield stress values, and that leads to this heterogeneity in the yield stress quantities. One might say that this is a bit system specific, which is a valid point. I think the idea that I'm trying to show here is mechanical heterogeneity emerges. There's a chance that because of the growth, assume you are doing experiments on actual soil, saturated soil. There's a chance that you get microbial growth, and the presence of those biofilms can also induce mechanical heterogeneity, maybe in the opposite direction. Maybe it increases the yield stress. The point is basically you get heterogeneity in mechanical properties, and that changes the motion of the bubbles, and that could lead to different shapes or different morphodynamics, if one wants to use a fancier word, for the colony.
Fantastic. Dan Bella asks about the second part of your talk. Does bombardment by the motile cells make the immotile core denser than it would be otherwise?
Yeah, that's a great, yeah. The answer is yes. So let me see if I can actually show. So you see this gradient in cell concentration, and then in the center of the core, the concentration is lower. That this length is basically determined by the diffusivity of by the lengths that the cells could travel before they lose their motility. So this boundary is where the last cell, the most the cells that travel the most is losing their motility and then sedimenting. So yeah, that's a great question. And that's why towards the end, you see that the concentration within the active annulus is actually decreasing. It's even more pronounced here because in the viscoelastic case, cells are able to travel even further into the core. So they're like a bullet that go all the way inside the core and then they sediment. Yeah, great question.
Okay, fantastic. There's another question. Sorry, I did not understand your experimental part. Do we have fast evaporation in your bacterial droplets? If so, wouldn't that create nutrient gradients from the interface of the core?
No. So there's no evaporation. In one of the experiments, unfortunately, we have even actually a little bit of condensation. So the droplet becomes a bit larger. But that happens in the span of 8 hours. So that's everything is slow compared to the dynamics of the bacteria. There's no evaporation. We
And
control the humidity.
perhaps in the same vein, because the first half you focus entirely on growth, the second on motility. Is growth and proliferation important at all, or do you see something different or interesting at longer timescales once?
In the bacteria case?
Yes, in the motile
Yeah, so it is important. I mean, it has an effect. I have done experiments in the case where there's growth as well. What you observe is before the doubling time, basically. If you
Mm-hmm.
go for lower concentration, you get a core. The core shrinks and disappears, and then after 30 minutes, 40 minutes, which is the doubling time of the cells, you get another core formed.
Mm.
And then the width of the annuals becomes lower, and that continues to become lower and lower because now you're basically at the maximum flux rate that is possible through the interface, but you are increasing consumption. You are increasing uptake, sorry. So yeah, it is important. It has an effect, but we try to avoid that because that causes more complexity.
Yes, yes, of course. Yeah. Um, Thank you. I don't see more questions in the chat. Are there other questions people would like to ask?
Hi, I have a question. For the second part in the experiment, I see that the density file across radius is not monotonic. Like for example, in some cases, surrounding the hard core, there is a layer of high density in the large concentrated droplet, and for the small concentrated droplets, there is a layer of low density surrounding that hard core. In your previous figures, which are not captured by the model. The model's concentration from the hard core to the surrounding are kind of drop smoothly. So do you have any idea of what might cause those?
Yeah, that's a great question. We tried to modify the model somehow to capture that. We were not very successful. My guess is there's some effect because of active turbulence. So especially in cases of higher concentrations where collective flows start to emerge, that definitely affects the aerotactic coefficient and aerotactic performance of the cells, which we did not include in this model because we wanted to keep it simple. So I'm assuming that could have an effect. Yeah, that's my strongest hypothesis, but I'm not 100% sure about it. But that's a great observation, actually. Yeah.
Okay, thank you.
I was wondering, coming back to this interesting connection you made about the merging of the colonies versus the typical demixing on the plane. Um, do you think in soils or in other heterogeneous contexts that the, that mixing between colonies produces an evolutionary pressure in favor of this kind of behavior?
An evolutionary pressure in favor of this kind of behavior. That's a that's a tough question. I'm not sure. Um, it's a bit difficult to answer because this is an isolated case with only one species, right? So it is hard to directly address anything ecological when you compare it with the soil because wherever you have these fermenters, you also have other bacteria that consume the carbon dioxide or produce carbon dioxide. In terms of especially for methane, there are methanotrophs, if that's the right term. So there are cells that consume the methane, and also there are cells that produce the methane. So for thinking in terms of evolution, really, especially ecological evolution, you need to consider those. So I don't really want to say something that is most probably not correct.
Okay, fair enough.
Yeah, but I mean,
Thanks.
the thing is that you can start to now play with these ecological interactions, right? You can start from two different species, and they could be they could be antagonistic, or they could be
Right.
mutualist, and then see how
Mutualistic.
they help
Yeah.
each other. Yeah.
Cool.
Yeah.
Great. I'm going to stop the recording now. Thank you again, Babak. We can continue with
Thank you very much,
questions after.
everyone.